All right, over to you.
Great, lovely. Thanks so much, Abigail, for that introduction. And of course, thanks for this prize and the opportunity to present my work here. So I'm currently a postdoc at UCL in a group of Yan and Mao. And the story I'm presenting today is a work that we're just about to submit that really looks at the role of 3D cell shapes and 3D cell contacts in signaling and developmental patterning. So let me just prepare my laser pointer here. So I'm very interested throughout my own work in developmental patterning and how in nature we have this amazing variety of beautiful but also robust patterns which are established in development. At the same time, I'm also very interested in how these patterns actually manage to stay robust when development is challenged by, for example, environmental perturbations. And so these ones here below are examples of cases where mechanical forces are actually influencing the developing tissue at different scales, at different time steps. And throughout all of this, I think it's a very fascinating question to ask how can robust patterning not only be achieved, but then also maintained when it's challenged by all these perturbations. And so for the purpose of this talk, the specific pattern I'm going to be talking about is a pattern of sensory organ precursors. So this is a pool of cells that is specified at the end of the third instar larval stage in Drosophila. And these cells here shown in red will acquire a proneural fate and then go on to develop and form sensory bristles which line the fly wing. And there are these beautiful sensory hairs that line all of the wing. And so this is actually quite a conserved process. So just as a reference, this is kind of the same process that underlies the formation of sensory organs in the inner ear in mammalian systems. And the way that we understand this patterning in the case of the fly is that at an early stage, there is an expression of winglets at the wing margin, and this drives a high expression and high activity of notch at the wing margin, which you can see down here in this notch reporter that very clearly marks that dorsoventral boundary. And so that from that central stripe, then two proneural regions are induced on both sides, as you can see here. And among those regions, then a process undergoes where cells signal to each other through notch delta signaling and lateral inhibition. And so the system sort of self-organizes until a pool of cells is selected among all of those to actually acquire the proneural fate and start then differentiating. And these are the SOPs, so these sensory organ precursors, which we typically visualize with, the marker neuralized. And so that's how the pattern looks like at the wing disc at the late stage. So you can see that we have these two beautiful rows of cells labeled in red. And what I also want to point out is because we have this high expression of winglets and high notch activity in the middle that practically insulate them, we can really think of the specification of these two rows as acting in parallel independently. So there, as far as we know, there is no signaling across that stripe, but only along the rows. And when we started looking at this pattern, one thing that we immediately noticed is that the pattern is quite sparse. So if you look at any given pair of these two red cells, you will see many examples where there are multiple cell diameters between the two cells. And this is important because in systems that are based on lateral inhibition and notch delta signaling, we know that the signaling range is very strongly linked with the spacing of the pattern. So if you have a longer signaling range, for example, the pattern is more sparse than what you get in the case of a smaller signaling range. So that's somewhat is indicating that there could be long-range signaling going on in this case. And to better understand this, we paired up with a team of Karen Page at UCL Maths and her student at the time, Francisco, who set up these beautiful simulations that are based on differential equations of notch delta signaling on a simple 2D model tissue. And they allow us to simulate basically how the signaling proceeds and then reaches a final patterning state on this sort of synthetic epithelium. And so in the case where we allow cells to contact and signal just to their first direct neighbors, we see that the simulations predict an almost perfect salt and pepper pattern. So there's an almost perfect alternation of SOP, non-SOP with a spacing of 1. And that's clearly not what we see in the experimental data. So if then Francisco actually allows the cells to increase their signaling range, and in this case, they're able to reach to 1.5 or 2 cell diameters away, then, as you can see, the simulations can recapitulate very nicely how the pattern looks like and also quantitatively what is the spacing of the pattern. So what this first result indicated to us is that at the wing margin, we are looking at some sort of long-range signaling that needs to go beyond the first neighbors in order to actually achieve the pattern that is experimentally observed. And this is something actually that is been seen in other systems that also are based on lateral inhibition. So for example, one case is the specification and refinement of SOPs in the notum of the fly, which occurs during the pupal stage, so at a later stage. And here it has been shown that cells actually use basal, very thin, long protrusions to extend their signaling range. However, in our system, we actually haven't seen evidence of basal protrusions play a role in the specification. But I also want to point out that the shape of the cells at this stage is dramatically different. So at the wing, in the wing disc, cells are pseudostratified, and as you can see from this animation, they're extremely elongated. And so our hypothesis was that actually, because of this unique cell shape, cells could actually increase their range of contact through contacts in 3D, which is something that had not really been looked at so far. And so just to illustrate this through a very simple sort of graphic illustration, the cells here are what we now define as a scutoid shape. So a scutoid is a kind of like a polygonal packing shape that can also change neighbors along Z, so along the apical basal axis. And the way that these could give rise to these long-range contacts is, for example, as you see here, if we take the red cell and we look at the apical surface on the top, this red cell could signal and potentially inhibit cells 1 and 2, the white ones. But because, again, these cell shapes can change and change neighbors across Z, when we move to a lower plane, we could then see that the red cell now not only signals to the 2 white cells, but is also in contact with the third gray cell. And so basically that's a simple illustration of how by changing the shape and changing the contacts, that red cell could now be reaching further than what can be seen from the surface. But of course, for this kind of contact to play a role in signaling, it would be important that both signaling receptor and ligand are present on these lateral surfaces. So we wanted to check that. And to do that, I imaged knock-in lines where either notch or delta are labeled, as you can see here. And what we saw is that, as expected and previously reported in literature, notch is highly expressed apically, but both notch and delta are also present along the lateral surfaces of the cells. And so any contact that cells have on the lateral surfaces could also act as a signaling interface. So we then wanted to actually look at these contacts in the context of the tissue, and that required quite a bit of work. So we spent a few years in the lab optimizing imaging and also analysis pipelines to now allow us to do single cell kind of segmentation in this extremely thick and dense epithelium. So I haven't mentioned this before, but wing disc cells are up to 50 micron tall. So they're extremely elongated, and that makes it really hard to image them all the way through. So I'm not going to spend a lot of time on the technicalities of this because we've very recently published this as a protocol on arXiv. So you're very welcome to have a look if this is something relevant for your system. But very briefly, what we've ended up kind of like optimizing as a final pipeline is to use 2-photon imaging as a way to really be able and penetrate deep through the tissue and image throughout all of the wing disc. And then in order to do the segmentation, we've actually created a pipeline that leverages several open access, very powerful tools such as CellPose, TrackMate, and Napari, and basically use them to have an AI-assisted segmentation that starts with pre-trained models and then steps to improve the 3D stitching of the cells. Because as you can see, our main struggle, which I think is the case with many kind of pseudo-stratified epithelia, is that these models struggle quite a bit in having a correct stitching in 3D of the cell. So that's usually the most challenging aspect. And we find that by including, for example, an ad hoc stitching step with TrackMate, that really helped us to get correct labels in 3D with minimal manual annotations. And of course, this is something I'm happy to discuss in more detail with the questions. But with this pipeline, what we can now basically do is take these 2D views of cell-cell contacts and now start looking actually at what the cells are doing in 3D. And when we started looking at these images, it was quite obvious from the start that we have many, many neighbor changes. So these are just some illustrative examples of cases where a pair of cells would not be considered as neighbors from the apical surface, but as you can see, they then get in touch even multiple times with each other deeper down along the apical basal axis. And so these are examples of these Z intercalations I was talking about earlier. So then if we quantify these Z intercalations throughout the whole tissue, this is how things look like. So cells apically start with an average of 6 neighbors, which is very typical for hexagonal packing in epithelia. And then as we go deeper and deeper in the tissue, they keep encountering new neighbors thanks to these neighbor changes. And what we saw was quite striking is that cells actually ultimately acquire twice as many neighbors as what they started with. And this is literally just looking at each new unique cell, which was not previously ever in touch with that initial cell. So this is a very remarkable expansion of the neighborhood that cells are undergoing. And I think it's also nicely displayed with this type of graphic where I'm highlighting a central cell and its apical neighbors in orange. And then what you can see in red are all the additional cells which deeper down in the tissue will become neighbors of the central white cell. And so what's clear, I think, from these images is that there is really this sort of isotropic expansion of the neighborhood that cells have, which also very nicely fits our initial prediction from the modeling that cells were increasing their range of signaling to 1.5 to 2 cell diameter range. That was kind of the range we always ended up with in the simulation. And this is what we see as well with this kind of extended 3D contacts. So then we wanted to better understand what the role of these contacts is on the signaling. And so for that, we went back to the modeling approach. And actually, Francisco introduced a completely new modeling framework, which we think is actually very helpful to think about signaling in a realistic tissue. So just to briefly explain, what we're aiming to do here is to do again, simulations of notch delta signaling equations, but have this done on a very realistic 3D cell topology, so realistic cell-cell contacts. And because it's quite hard to mathematically form those extremely elongated shapes with all of those neighbor changes, what we decided to do is to create simulations that run based on experimental data. So we start with the segmented data, and we use that to create a network of cell-cell contacts. So you see we have the centroid of the cells, we have information about cell-cell contacts, and that creates sort of like a network where we can then let the simulations run and let them predict where SOPs will form. And then, of course, we can measure the spacing and compare the spacing with the experimental data. And just to highlight that the main difference in this case compared to the initial 2D model is literally how we calculate the total neighboring delta activity for each cell. So in this case, we have to consider every single layer. So we have a sum over all of the Z layers. And then on top of that, for each cell, we sum over all of their neighbors. And what's also really nice is that in this model, we're inputting, again, real cell-cell contacts. So we have information such as the shared edge length between each pair of cells, which is important because signaling is proportional to the sort of shared interface between cells, and as well as the perimeter of the cells. And then a key advantage of having such a modeling approach is that it allows us to explore and test the role of different parameters. And so one of the, I think, most important things we wanted to check is what is the role of the strength or weighting of the signaling in the apical versus in the other planes. Because as you've seen from my staining at the beginning, we know that notch is enriched apically. So how much signaling do we need laterally to kind of make a difference, right? And this is something that can be explored very nicely in the model because we can arbitrarily change, for example, how much weight we give to the apical contacts compared to the 3D contacts, and then see the effect of that. So as an example, these are sort of like 3 example cases, and it's just a small zoom in on a small region of the cell-cell network. But it's just to show you that we can get simulations where, for example, there is only apical signaling. So we are weighting everything towards the apical layer versus on the opposite case where we're weighting everything laterally. And the difference here is that, as you see, it changes the cells that are predicted to be SOPs. So it changes also how packed the pattern is and the spacing of the pattern. And so from that, we can then kind of like attune the signaling strength. And what we found here is that as long as we have at least 20% contribution from the lateral surfaces, that is sufficient for the model to predict that there's a switch between the salt and pepper spacing to more sparse patterning. So that has been actually quite a valuable tool to really explore the role of these 3D cell-cell contacts and understand what are the requirements to get sparse patterning. But at the same time, we also wanted to have an experimental test that the 3D cell shapes could affect the spacing. So we looked for conditions that would allow us to alter the shape of the cells and therefore alter the contacts. And we found that when we increase myosin activity in the pouch of the wing disc through an MBS RNAi, which is a condition in the lab we have previously characterized to make the cells also stiffer. So this sort of straightens the cells. You can see looking at the lateral interfaces here, for example, compared to the control, that cells are much straighter, as you can see also from the 3D reconstructions. And so does that correspond to less Z intercalations, as one might think? Well, for the control, we know that, as I've shown you previously, the cells have many, many intercalations, and on average, they tend to acquire 5 new unique neighbors in Z through these intercalations. However, in the straightened cells, we see that that's much less. So in the straightened cells, the cells on average acquire only 2 new neighbors through apical basal intercalation. So indeed, they are straighter, they have lost 3D contacts, and therefore they have fewer neighbors. So then we wanted to check the effect of this on the patterning. So we looked at the SOP specification in the control versus in this mutant background. And what we could see, you can see already from the image, but also by quantifying it, is that very clearly in the straightened cells, we get a pattern that is tighter. So there's the distance between SOPs is much less. And so this is exactly what we sort of would have predicted, because by changing the shapes, we have lost 3D contact. So we've basically shrunk the signaling range of the cells. And because of that, we predict that the spacing would be smaller, and therefore the pattern would be more tightly packed. And so just to summarize what I've shown you, we've seen that the cells at the wing margin need to be able to signal beyond first neighbors to see the experimentally observed pattern of SOPs. And we have developed this new imaging and segmentation pipeline to allow us to look at the contacts, and that has allowed us to find that the wing disk cells actually increase contacts by almost twofold. So they contact twofold more unique new cells in 3D compared to just apically. And that the topology of these contacts is really essential to set the inhibition range and control the spacing. And when we genetically straighten the cells, then that decreases their range and results into a tighter pattern. Yeah. So with that, I just want to thank everyone in the lab and the students I've been lucky to supervise in the past few years. Quite a few of them have been involved in improving the segmentation pipeline and doing corrections on the images, so I'm really grateful. Many thanks to Baz, who co-supervises me on this project, our computational collaborators, Karen and Francisco, that have really been working with us hand in hand for the past few years. And many thank you for your attention.
Thanks very much, Giulia. That was absolutely beautiful. I managed to drop out with my Wi-Fi right at the end, as I had a sudden panic, but all good, and I'm glad it wasn't your Wi-Fi that had a problem. So can I start you off with a question, which is, as you said, these cells are really tall, and I'm wondering, is this a rare type of cell shape, would you think? And this ability to contact lots of different cells? Or are there other cell types that you can think of that have this sort of really thin shape?
Yeah, it's a great question. So indeed, like scutoids have been discovered only a few years ago. It's a relatively newly described shape. But having said that, I think they have been now observed in more and more systems. It tends to be where there is a curvature. So lots of tissues that are curved, for example, salivary glands or like tubular type of tissues are usually scutoid. Maybe not to the extent that these cells are. So in the wing disk, I've quantified that basically 100% of the cells will be scutoid. And as you see, they undergo many neighbor changes. But again, I think it's quite a prevalent shape. You might be looking at just a slightly maybe lower rate of neighbor changes. But I think whenever there is, let's say, a sort of like curvature, it tends to be quite a good shape as a mathematical solution, basically to pack the space. So they're quite prevalent across different systems, maybe not just as extreme.
Managing to unmute myself. And we've got a question in the chat. Just a reminder to people that there's a Q&A box at the bottom that you can use. So the question is, in the image of the SOP pattern shown in red, the SOP cells seem much larger than their neighbors. Is cell size linked to the patterning process?
Mm-hmm. Yeah,
they said clarifying they're talking about the sort of 2D images.
Yes, absolutely. That is a great question, and I would say yes and no. So the reason it's a very good observation, and the reason that the SOPs you see are bigger is because again we're using here a relatively late marker of fate. So by the time you're seeing them all nice, bright and red, they're really fully committed, and we know that they stop the cell cycle. So they stop dividing and therefore they grow. This is something we have observed and we've been able to quantify also in the 3D. But this doesn't seem to be the case at earlier stages. So what we see is really that the increase in cell size comes after they kind of like specify, because they stop dividing. And this is kind of also what the model predicts.
Great. And I had one other question, which was about the myosin. And so you've got these sort of thicker cells that are straighter. And so the question is, are they also shorter? Have you reduced the length? So have you got is the total volume the same, or have you actually sort of increased the volume of your cells?
Yeah, that is a great observation. And I mean, of course, in an ideal world, you would want to find the perfect perturbation that only changes, let's say, one feature without affecting anything else. In reality, what is happening is, yes, we think that there is sort of volume conservation. it seems that the cells divide a bit less in that condition, so you end up with slightly larger cells. Interestingly, the apical packing is identical, so they have exactly the same distribution of roughly 6 neighbors apically. They're just bigger, so it kind of scales in that sense. And they are also slightly shallower, so in a way they kind of become flatter and a bit wider. And so that's one thing that we are also checking with our modeling. Again, we can sort of input the perturbed topology and see the effect of that on the spacing. But the important thing is that the apical packing stays the same.